studying sensory systems is a really unique aspect of biology because, every creature sees the world in a different way. We see the world in a different way than a honeybee or a snake, and that really depends upon the biophysical and molecular sensors that we have in our bodies that interpret all these signals from our environment and then convert this information that our brain can kind of reconstruct as our world.
Over the last few decades, we have learned a lot about how we see, smell, taste. Of course, smelling and tasting is all based on chemical sensing, and this is actually the language that most of our cells in our bodies use to communicate with each other. But the somatosensation, which is sense of touch, pain, proprioception, etc., all of these depend on sensing physical stimuli, which is very different. So how do you sense temperature? How do you sense pressure? And translate this into a language that neurons understand, which is an electrical signal, was not known. It was known that these specialized proteins called ion channels exist, but their identity was not known. So my lab has found ion channels responsible for pressure and earlier work in temperature. What your skin is sensing is the indentation. So anytime you touch, your skin is being indented. What's really fascinating is we say there's the sensor in the skin, but actually most of the sensing is happening at this very, very thin, very long projection of a sensory neuron that starts in the back of your spinal cord. This neuron sends processes everywhere in your body, including the skin. And at the tips of these neuroids, that are sub-micrometer in length, sits this tiny, tiny ion channel, which is in nanometers length, that when the plasma membrane, which is the fatty surrounding of a cell that protects the cell, there's this little protein sitting there. And this protein is, as you can see here, is a molecule that comes with 3 identical members in different colors. They come together and sit in the membrane shown in this green color, and they actually bend the plasma membrane. This piezo-2 protein, there's 2 of them, we call them piezo-1 and 2, are proteins that are encoded by DNA, and so a gene. So there's this gene that its only role is to make a protein that sits in a few cell types on the membrane, and its only job is to sense pressure. And when this cell gets pushed or dented, they flatten. And when this flattens, conformation of the protein changes and ions start flowing through this protein. And when that happens, you have the first signal that starts the process that we call touch.
What molecules in our body react to heat and cold?
At least as far as we know, most of these molecules belong to the same family of membrane proteins, these ion channels called trip channels, which are actually first discovered in flies, in fruit flies. But in mammals, a number of them, different ones, and we don't know yet all the ones that contribute to temperature sensation, but at least 2 of the ones that we've looked at, the receptor for chili peppers, for capsaicin, and the receptor for menthol, these TRPV1 and TRPMA channels, they're sort of molecular cousins in the same gene family, and one of them senses heat and one of them senses cold. This is a model of the capsaicin receptor. This hole in the middle opens up like a donut hole, and it allows ions to flow down their what we call electrochemical gradients. So outside there's a lot of sodium ions, and the sodium ions rush into the cell, and the currency of communication in the nervous system is electricity. So when ions are flowing across the membrane like that, it generates an electrical current, and then this excites the neuron. That initial excitation is then carried along the neuron, say from your finger all the way along into your spinal cord. That information gets relayed to another neuron that carries that ultimately to your brain. Your ability to recognize something hot or cold is determined by the biophysical properties of this protein, which is really kind of a magnificent thing, right? Our route to discovering that was a little bit indirect in that we didn't go in first asking what are the molecules that sense temperature. We asked what are the molecules that sense these pungent agents, these natural products, menthol, first capsaicin and then menthol. And with capsaicin we identified this channel, and we knew it was going to likely be important for some aspect of pain sensation, but we didn't really know what the endogenous normal activator, physiologic activator might be. So we threw all kinds of things at it that we thought made sense in the context of pain, like neuropeptides, neurotransmitters, and didn't really find anything that seemed to be consequential. And so then we decided, our pain system does more than detect chemicals. It detects physical forces like touch or temperature, and so we tried changing temperature. Cold didn't do anything to the capsaicin receptor, but heat did. And so we realized that that the capsaicin receptor and the heat receptor were the same. And in retrospect, of course, everybody thinks we made the connection right away because you eat a chili pepper. But it didn't have to be that way. It could just be that they're on the same cells and you activate the same pathway. And so that was kind of a eureka moment when we realized that this is kind of a very parsimonious explanation for your everyday experience. Among sensory systems, I always say, I think the somatosensation and pain is probably the one that's most important for our survival and well-being. Because if you don't have it, you really have trouble learning how to avoid things that can be injurious.
Ladies and gentlemen, please welcome to the stage David Julius and Ardem Patapoutian. I've got about 9 million questions.
I know, yeah, so many questions, yeah. But I want to start with just sort of, as someone who's not in this field at all, we think of our senses as quite well understood in a way, our sight, our hearing, and things like that. But actually, some of our senses have almost sort of been neglected. And you've both been involved, as we've just heard in that amazing film, in understanding how different signals are interpreted by our brain and our subsequent reaction to them. So, Ardem, I'm going to start with you. Just tell us more about that.
I think the complexity of somatosensation begins with location. So for visual sensing, of course, we know where our eyes is. It all starts there, the nose, the mouth, etc. So somatosensation is really all over your body. And unlike, again, smell, which is just chemical sensing in a way, we have temperature sensing, we have mechanosensation, both we perceive as touch and pain. We have proprioception, which is one of my favorite senses, mainly because people don't even know they have it, and I think it's one of the most important senses. And this is how you know where your limbs are in space without looking. So you take this for granted, but I can easily close my eyes and touch my nose. And this ability is based on the same type of sensory neurons we're talking about, but they innervate muscle. And from how much muscles are stretched, you've learned ever since you're a baby to know what kind of space you occupy. And so I feel like it's a very basic physical consciousness of what space you occupy, but also allows you to do things like stand up, walk. So again, you take these for granted, but without proprioception, this sense that most people don't know about, you wouldn't even be able to do these tasks. And there's actually people who have mutations in these genes that we've worked on. Alex Chesler is here in the audience, who's done much of this work, is analyzing that these individuals don't have piezo and have a very hard time simply walking, standing up, and all these tasks. And so fascinating group of sensations all bundled under somatosensation.
Yeah.
And always, if a policeman stops you in the car and asks you to close your eyes and touch your nose, you're in trouble.
Yeah.
David, how did you get into this? Because I think it was via some substances that were probably rather common around Berkeley at the beginning of your career.
Yeah, I started my graduate career, doing something very different, working on using yeast genetics to understand basic cellular processes. And I did at one point get interested, probably because of the environment in Berkeley at the time. I won't say what years those were. How we interact with the world in terms of pharmacology, let's put it that way, and natural products. And I began to become fascinated by understanding how hallucinogens work. How does things like psilocybin or LSD...
And of course, that's come
alter...
right back round again, hasn't it, now?
Yeah,
it's hugely popular.
That's right. Yeah, now it's microdosing and all with...
Yes. So
psychotropic.
you took those, and then how did they give you an insight into sensory perception?
Well, that wasn't, in those days, I wasn't really interested in sensory perception so much as it's what really got me interested in neurobiology. But there were two things about that. One is it sort of kindled my interest in neuroscience, because we knew that hallucinogens must work, in particular on serotonin receptors in the brain. So my first foray into neuroscience was looking at serotonin receptors, identifying genes that encode those. But it also kindled my interest in this whole idea of using natural products to explore pharmacology in the brain or the nervous system. And understanding, I think that's really sort of a fascinating nexus between human behavior, chemistry, and physiology. There's so much to discover and uncover there. How do people discover natural products? How do they learn to use them in their everyday life, ritualistically, or for medicine, or for spices and culinary purposes? And then how do chemists identify the natural products that underlie those sensations or those activities? And then how do physiologists use those chemicals to understand how they work in the nervous system or the cardiovascular system and exploit that as kind of a full circle of human behavior going around to biology? And
capsaicin is a really interesting one to start with. Now, are you one of those Americans that eat very, very, very hot chilies?
No.
All right.
I like spicy food, but everything in moderation. But so capsaicin had been known for many years to be from work that was done in the 50s and 60s from Hungarian groups to be a very potent and selective activator of a subset of neurons in our body, the ones that Ardem talked about, and that play a role in somatosensation. And in particular, in the somatosensory system, there are multiple different types of nerve fibers, and there's a subset of those nerve fibers that are dedicated to detecting what we call noxious stimuli and that initiate pain signals. And capsaicin was known to be a very potent and selective activator of those nerve fibers. So we knew for many years that if we could understand how capsaicin worked, we would likely uncover some important basic principles about the molecular aspects of pain sensation. So that was kind of a goal in the field that hadn't been reached, and we decided that we would look at that.
Amazing, yeah. Ardem, can we just explore your background a bit? Because as I understand it, you were in Beirut having a pretty tough time, actually. You were held prisoner by armed militants, and you leave Beirut and you go to the US and get some sort of scientific experimental experience. And whilst David's working on the TRP receptors, then you become interested in, is it how the pressure receptors actually work, and that sort of translation from mechanical into electrical, is that right?
That's right. So yes, the question, our lab worked on the TRP channels and temperature sensation as well, but we got really interested in kind of the second arm of somatosensation, which is very relevant for both touch sensation and pain sensation. Both can be mechanical in nature. And we, like many others, were trying to figure out how do you find these ion channels, because they're so rare and small, they're not located in one place. And one of the cool things that happens in basic science is you have one question, and this kind of theme was talked about earlier today, is you're driven by curiosity, and it just takes you in all these kind of directions. Yeah. So you keep saying it's pressure sensing, which is correct, and that's very important for touch. And after we found the molecule, looking at other parts of the body, we've found fascinating roles of mechanosensation that we didn't think about before. And so the same molecules that are involved in sensing touch appear to be involved in sensing blood pressure. And at the same time, when you have to go to the restroom and you feel like you have to go, bladder filling is a... So in retrospect, you would say, of course, that's pressure sensing. It's about stretch. And so it's kind of an awesome place to be in our science right now, because a lot of the projects get initiated simply by intuition of a postdoc. Right. For example, someone came into my office and says, the uterus expands 30 times during pregnancy, and there's not much literature about how this is actively sensed. And so this is one direction we're going into with massive implications for health, of course. And we've gotten all kinds of surprises. Red blood cells sensing pressure. And one of my favorite examples is just last year, we had a paper that showed that the same molecule in plants exists, and it actually is in the root tips, sensing how hard the surface is that they're going through. So the idea is that plant roots also need to sense their environment and navigate accordingly. If the surface is too hard, they need to move away from it, go around it. And he found that this molecule is involved in that process. So just imagine that we've evolved to use the same type of molecules to sense touch, and for plants to sense how hard the surface is that they penetrate. So that gives me pause, and I kind of really love that idea, how evolution works.
That's incredible.
So what's fascinating, though, is that the same level of touch can mean so many different things to a human. So you can have a touch on your arm which is about comfort, or about reassurance, from a mum to a child. It can be, confidence boosting. It can be sexual. It can be all of the above. So there are so many different things. How is that distinguished? What's going on? I mean, is it other sensory information that's coming in that's making us interpret touch in different ways?
So this is actually an active area of research, and one of the predominant ideas is that we call it a labeled line idea. So it's actually not the molecule, it's the cells that mostly determine. So there are neurons that are specialized to sense pain. Even within touch, there's some that sense discriminative touch, which is being able to realize I'm being touched by two different fingers, to what we call affective touch, which is the pleasurable touch that gives us, important for our emotional health, etc. And so, although it's the same molecule, seems to be involved in many aspects of touch, they're actually distributed on different neurons that each specialize in giving very specific signal. And so this all comes together of what we think of sensing touch, which is of course all happening in the brain, but in the periphery there's all these wires. Imagine this complex group of wires, blue and red and white, and electricians would know what is each doing, and we're trying to figure that out. But some of them all express the same protein that we're talking about.
So how do you know? So if you put your finger in a flame, what are you feeling? Do you feel heat or do you feel pain?
David can take this too, but again, this is an active field, and there are some that are what we call polymodal nociceptors. Complicated two words. Polymodal means it can sense many different types of damage, and nociceptors means damage sensing. We distinguish nociception from pain because pain is almost an emotion in the brain, while nociception is this peripheral neuron sensing noxious stimuli, things that are not good for you. And so when you're putting your hand on a hot stove, many things are happening. The TRPV1 expressing neurons are firing that tells you about pain. At the same time, heat signal is coming. And so many times, of course, you don't sense stimuli one by one, you get many of them coming in at the same time. It's the processing of all this that is the sensation of pain. And one of the actually great applications of the work that David's lab has done and ours is finding these receptors gives you actually tool to start answering the questions that you're asking, which is many people are interested in what's happening in the brain while you do this, if it's noxious or innocuous. And if you have these molecules, then you have tools to label these different neurons and follow their activity in the brain.
So David, if you put your hand in a flame, you take it out pretty quickly.
Yeah.
And that's I guess, one of the purposes of pain, to stop you doing stuff that harms you. But what happens in chronic pain? Because chronic pain stops being useful. In fact, it becomes an enormous hindrance, and chronic pain is at the basis of, the opioid epidemic, I suspect.
So there's, different types of chronic pain. I think that's one of the big questions is, we always use this word pain as one word like we do cancer, but chronic pain is a multitude of different problems. There's migraine pain and musculoskeletal pain and irritable bowel syndrome type, visceral pain. And I think one of the goals is to understand how they differ molecularly, what different cell types are involved, and what's going on at a biochemical level. And I should say that some of the channels that we've worked on, whereas the piezo's are very dedicated to sensing stretch, these trip channels, while they detect heat or cold, they're also a little bit more polymodal because they can be modulated by different factors that get produced when there's injury, and most particularly inflammatory injury, and contribute to inflammatory pain. So in addition to measuring temperature per se, they're also being modulated by different components of what we call the inflammatory soup, like change in local tissue pH is is largely associated with tissue inflammation. Prostaglandins and all these other things that get produced that we know exacerbate pain, they act on some of these channels to enhance their sensitivity. So for example, if you get a sunburn and you would go into a shower that normally you would experience as being very comfortable, you might jump out because you think it's incredibly hot, excruciatingly hot.
Yeah.
And that's because all these inflammatory mediators have worked on these ion channels, interacted with them, and shifted their thermal threshold, so now they're being activated at a lower temperature, closer to your skin temperature. And so when you get in the shower, or just normally, you might feel pain, and even when you get into a shower that has a normally warm, comfortable situation, you perceive that as being now impossibly hot, because you shifted the thermal sensitivity of these ion channels and therefore of your sensory system. So these machines are very interesting machines in terms of being able to integrate not only what's happening in the tissue milieu and readjust the gain of the sensory nervous system, really to tell you that you've injured yourself and you need to guard it and protect it. And so we're interested in, and people in the field, in what's happening locally, for example, when there's injury in the nerve fiber in the tissue, that then modifies some of these ion channels like TRPV1 and others to exacerbate their activity and initiate the process of chronic pain by leading to pain hypersensitivity. But chronic pain can be engendered at a lot of different regions in the nervous system, and once you have, for example, very repetitive or exacerbated activation of the nerve fiber, you can then change the wiring of the system at the level of the spinal cord or brain to basically instantiate these long-term, what we call maladaptive changes. So this is a big challenge in the field to understand where these changes occur in different places, what's driving them peripherally, how they become manifest centrally, and how this leads to different types of chronic pain syndromes.
Now, we hear of people who they have a high pain threshold, and we also hear of people who don't feel pain because they have a genetic condition, which means that they can't, and that's very dangerous for them. But is there such a thing as a high pain threshold, and is that mediated by more or less receptors?
I think people. Clearly, we know from our own experience that under different circumstances or among different people, we have different thresholds for pain. But, pain is a complex, as they say, no brain, no pain. So, the sense of detecting the stimulus peripherally is nociception, but you really don't understand that cognitively as a pain response until it's integrated by the brain. So there's a lot of processing that goes on there. And I think that people's, thresholds for pain are clearly modulated by
like emotion, because
like emotion,
yes.
Yeah. There's an emotional component, there's a sensory component, and so different pain levels there are some people who have differences in terms of their peripheral sensation, and a lot of that's also going to be cognitive. What you can deal with, what your experience is for pain, and what the context is. If someone's expecting something, it's very different than if they're not expecting something, how they feel pain. So there's a lot of experiential processing as to how you feel pain and under what circumstances.
Ardem, why neuroscience? And why this particular area, I guess,
why
as well?
Not?
But, what is it about this that really motivates you and you get so passionate about?
We heard early this morning, even astrophysicists want to be neuroscientists now. So, I think the study of our own bodies, I think you guys mentioned this too, is just the fact that now technology is getting better and better to study this at a more detailed, more clear molecular level is absolutely fascinating. Again, when I think about evolution, of how over this long time that we've evolved, these machines of molecules evolved to make us help sense the environment and how we process it, is absolutely fascinating to me. Specifically for neuroscience, as a graduate student, I was not a neuroscientist. So I was actually looking for fields to get into, and I was looking for something that was complex, and at the same time has health relevance and lots of open questions. So I was actually considering neuroscience and immunology, and I decided immunology has more jargon than neuroscience. I don't know if that was true or not, but I picked neuroscience. And the reason I got into the peripheral nervous system, which we keep talking about, is it's a bit intimidating for a graduate student to get into this new field. And I thought the peripheral nervous system is so nice and simple. So I said, I'll start there as a postdoc and then migrate to the brain after a few years. And 8 years later, I'm still in the peripheral nervous system because there's still so much to discover.
Yeah.
But yeah, I think biology is an amazing field. It's maybe we've had a few golden eras, but I think this is certainly one of them, and it's a great future and great science to be done as well.
And it wasn't just LSD that presumably prompted you into neuroscience.
No, I mean, I think that's, I think I gravitated towards neuroscience and to sensory systems in particular because, neuroscience, because at the time that I started getting into the field, it was really the birth, what I would say, of molecular neuroscience. It was really when people started to apply genetic methods to trying to understand the mechanisms, molecular mechanisms that govern neurotransmission. And in particular, everybody was trying to clone genes for different types of ion channels and receptors and understand all this pharmacology and really sort of get some handle around the sorts of molecules. Like serotonin, we know for many, many years is a major transmitter in the brain. Yeah. It wasn't clear how many different receptors there were, how this would work, what was the genetic basis for the receptor type diversity, how could you have some drugs that had certain effects on serotonergic systems and not others. And so it was really just this wide open area that seemed ripe for somebody like me who'd been trained in molecular biology and biochemistry to sort of jump into and really sort of try and get to the genetic roots of that. But I think for me, sensory systems are really fascinating because it's really, as I said in that little video, how we see the world around us. The world to some degree is kind of a subjective thing in a sense that it depends entirely on the spectrum of colors that our eyes are able to see in the electromagnetic field, what kinds of temperatures we can experience, what kind of odorants we can smell, and this is different for different animals on the planet. And so our world appears very differently to us than, for example, a snake that can see in the infrared range, or a honeybee that can see in the UV range of the electromagnetic spectrum. And so, how our brain interprets the world is completely dependent upon the properties of the little molecules in our nervous system and in the membranes of these cells that take in all this sensory information, all these stimuli, and then activate the right types of neurons. And so, what could be more fascinating than understanding how these detectors in our body sculpt our mental picture of the world around us?
Yeah.
And it's also, as Ardem said, this area, no matter what sensory system you study, has tremendous relevance to medicine and physiology. And for me, pain really seemed to be, it was one of the sensory systems about which we knew the least when we started working on this. And I've always felt that it's one that has the most promise for medicine, because we all know that we need to deal with pain in a different way and in a more complex way. And we need some inroads to that. And I think that you can get lucky and develop some drugs or therapies, but really it's going to be based on curiosity-based science that tells us about mechanism, basic mechanism. And until we have that foundation, it's really hard to start making rational approaches to developing new types of pain medicines.
Yeah. I mean, what you're both showing in spades is that glorious thing about science is that you open one door and then 7 other doors open in front of you and another, 30 in front of that. So it's just a constant,
Yeah,
it's a constant
You never
quest.
put yourself out of business.
Indeed. But what comes next? So Ardem, I think you're doing something on fat cells.
Yes. Before I answer that, I also wanted to just point out that we've very privileged to get this recognition. But I just want to have a shout out to all the trainees in our labs that do all the work. When we talk about what we did, my lab is usually 12 to 16 people of graduate students and postdocs who actually, do the work, who become the next professors or people who work in the pharmaceutical industry. And I just wanted to say a shout out to Bertrand Coste, who's here, who actually cloned piezo, the channel that we're talking about. He's a professor in Marseille now, and I just wanted to mention that. But yes, as David said, curiosity means you never get out of business. There's always something else to answer. And as I kind of hinted at, our lab is now taking this pressure sensing and seeing what other biological processes and diseases these are involved in. And even in fat, now we know that these mechanosensors are also innervating fat tissue, and we have no idea what is it in fat that there is mechanical stimulus that's being sensed. And so we don't have any data on this yet, so that's how you guys are hearing the very most recent and future stuff. But if you have any ideas, come and tell me. But we're trying to figure out why is piezo neurons expressed and innervate fat tissue? What are they sensing, and what do they do with that information?
Fascinating. What's next for you?
I think we're sort of pursuing 2 lines. One is sort of going a little deeper in terms of more reductionist stuff, and then one is sort of growing a little bit larger in terms of physiology. So in the sort of more reductionist things, now we have these molecules that we've studied, like piezo or TRPV1 or TRPM8, and one of the goals in the field, and one of the things that I've done, for example, with a colleague, Yifan Cheng at UCSF, is to use this technique that we've heard a lot about in previous talks, electron microscopy, in this case, cryo-electron microscopy, to really see what these molecules look like in near atomic detail. And that helps us understand, for example, how they respond to things like pressure or temperature, what happens to the molecules, how they respond to these forces, how they open up, how they're regulated. And then ultimately, you can look at all the nooks and crannies in the molecule to understand how you might design drugs to modify their behavior, or how inflammatory agents like prostaglandins and protons modulate them to change their threshold for temperature sensation, for example. And then on a more sort of broad level, we're trying to understand this question, make inroads into this question I brought up about how are different chronic pain syndromes different. And the goal is to really to pick 1 or 2 that you're most interested in and really dig down and try and understand the molecular basis of different types of syndromes. And one that we've worked on recently, actually together with my wife, who's here, Holly Ingram, is to try and understand what are the factors that are involved in visceral pain, like pain that's associated with things like inflammatory bowel syndrome. What are the molecules? One of the molecules that we've worked at, this TRPA1 receptor that we both worked on, we call the wasabi receptor because it's activated by pungent agents, and wasabi is found in cells in the gut, and we know that that's important for signaling pain and irritancy in the gut. What are the nerve fibers that are involved in that? What are the cell types in the gut are involved in that? What kinds of chemical factors modulate that? And also we know that there are big gender differences in that sort of aspect of pain. Women report issues of GI pain more frequently than men, about 2 or 3 times more frequently. What's the basis for that, physiological and genetic basis? So there are pain models like that that are very ripe for mechanistic insight, and so we're trying to look at a few of those to really gain some specific knowledge about them.
Just amazing.
It's amazing. I'm completely hooked.
Absolutely amazing.
We've run out of time.
Yeah, thank you so much for a truly inspirational conversation there. It was so, so nice. Thank you both. Please do congratulate you both again, David and Ardem. Congratulations. Thank you. Thank you very much.
And I'm just going to talk to the camera directly online. If you are one of the trainees of Ardem Patapoutian, you are much appreciated. Thank you.